Okay, we should get started. Greetings to all of you and welcome to the ZOOMinar today. Today's speakers are Dr. Raz Jelinek from Ben Gurion University, Israel, and Dr. Claudio Luchinat from Florence, Italy. I also would like to welcome Dr. Enrico Ravera from CHUM, Florence, Italy, to the panel. So today it's my pleasure to introduce the first speaker, Dr. Raz Jelinek from Ben Gurion University. Raz has a bachelor's degree in chemistry from Hebrew University, Jerusalem, and a PhD from chemistry department, UC Berkeley. And then he did postdoc at UPenn in the Opala lab, where I overlapped with him for 3 years, working together. In fact, even we published a paper in JACS. That's great, Raz. Then Raz joined the Faculty of Chemistry at Ben Gurion University, Israel, where he is a professor now and also Vice President Dean for Research and Development. Raz is a very interesting and dynamic person. He cannot rest, so it's no surprise that he works on so many areas. His research spans nanotechnology, surfaces and thin films, sensors and biological membranes, and protein misfolding and aggregation. Raz's excellent contributions to teaching and research have been awarded with many honors. These include the 2009 Toronto Prize for Research Excellence and the 2020 Quality of Teaching Award at Ben Gurion, and the 2018 UK-Israel Science Lectureship Award. So with this brief introduction, I'm delighted to welcome Raz to give his talk. Raz, it's all yours.
Yes, thank you very much, Rams. Pleasure to be here. Even if after 2 years, I'm quite fed up with the Zoom lectures. I couldn't say no to Rams, and this is a great initiative. And even though I don't see anybody in person, I think it's a very nice venue for science interactions. So I'll tell you a little bit about some research project that we have recently. It's actually a story, a nice story. And who knows, it might also shed some new light on amyloid diseases and particularly the role of amyloid fibrils in disease and normal physiology. Okay, so let's start with this slide, this introductory slide that we probably all know about very well. Amyloidogenic diseases are very diverse, and what we know about all these diseases that monomers, protein monomers, for some reason, some of them known, some of them not, form fibrils. Here you can see the list of different diseases that have amyloid, that have formation of amyloid fibrils. The question is obviously if there is some common theme among all these diseases and all the pathologies of these diseases that are maybe related to the fibrils. And this is actually a still open question, even after decades of work on amyloid diseases. We are not completely sure. We're actually not sure at all about the exact reason of toxicity of these diseases, particularly what are really the roles of amyloid fibrils. And some theories, some hypotheses talk about membrane damage, which actually is not induced by amyloid fibrils, but rather by profibrillar species. There are models of oxidative stress that might play a role, and amyloids might contribute to toxicity of amyloid diseases. There might be also effects on energy cycles in the cell, for example, in mitochondria. As you see, it's still a black box. Let's shift gears a little and talk about catalysis, okay? And specifically catalytic peptides. Here you can see a scheme of the active site of an enzyme, okay? And what we all know about active sites of enzymes, that you have a certain organization of amino acids that take up the substrates and catalyze certain reactions, certain biological reactions that do something to the substrate, usually break it up or do other redox reactions. In recent years, actually not so recent, but in last few years, several groups have shown that you can have catalysis or enzyme not just in enzymes, but in peptides. And not just peptides, but peptides that undergo self-assembly. Some of these examples have involved peptides that form amyloid type aggregates. Here you see an example of a recent paper showing a short sequence containing histidine and serine that form aggregates, self-assembled species. And these self-assembled species have shown catalytic activity. There have been actually quite a few very nice studies on catalytic activity of peptides. Here you can see a recent paper by the Lin group from Nature Chemistry. There are other examples of other short peptides that form self-assembled aggregates. Some of them can be described as amyloid aggregates. And these amyloid aggregates are catalytic. It's actually a very interesting phenomena. It has been related to early life studies. Some people suggested that these peptide aggregates contributed to the formation of the first generic, let's put it that way, generic enzymes. So that's an emerging field, very interesting. The thing with this field is that most studies, most examples of catalysis in self-assembled peptides or amyloid peptides have been done with synthetic peptides. And even in almost all studies have used cofactors, mostly metal ions, to induce catalysis. What was our question? Okay, so our hypothesis was that physiological or natural amyloids are also biocatalytic. Not only that they're biocatalytic, but we hypothesized that natural amyloid fibrils catalyze important biological reactions that also play roles in disease pathologies. The idea here is that similar to enzymes that form the active sites in a certain pocket formed by the three-dimensional organization of the enzyme molecule, what we believe or what we hypothesized when we started this research, that there are catalytic sites on the fibril surface. So the fibril surface provides a template, or the pocket, you might call it, for the occurrence of catalytic reactions. So this is what we studied, or we hypothesized in the research. We have a monomer, the monomers of amyloidogenic peptides, forming oligomers. The oligomer is going to form fibrils. And then on the fibrils, we're going to have, in a certain organization, catalytic sites. So here you can see schematically the catalytic site that essentially catalyze, in this case, a breakage or disintegration of a substrate to a product. Let me show you the results. Okay, and the results involve a well-known protein probably the most well-known protein in the amyloid world. The beta amyloid protein, the 1 to 42 sequence of the beta amyloid, well known as the most likely the toxic species in Alzheimer's disease. If we look at the sequence of 142 beta amyloid, it has several areas. The first, the 16 amino acid in the N-terminus are believed to be largely unordered. Then there's a beta sheet domain, a loop domain, and a hydrophobic domain that is primarily involved in a beta sheet in fibril formation. So the experiment was pretty simple. We took the monomers, we incubated them until we formed the fibrils, and then we asked ourselves, what's going to happen if we're going to add substrates? The substrates were added both to the monomers and to the fibrils, and the question was quite simple. Is there going to happen a catalytic reaction in this case? The first reaction that we studied is a very well-known model, biological hydrolysis reaction. This is essentially the catalytic reaction of a polynitrophenyl acetate that is hydrolyzed and form the polynitrophenol and the acetate unit. Both NPA and polynitrophenol have color, visible color, so it's very easy to follow this reaction using UV vis absorbance spectroscopy. So here are the results. What you can see here is the formation, the velocity of formation of the product, the PNP product, the polynitrophenol product, as a function with addition of beta amyloid, the 142 beta amyloid, either in the monomer form, this is in blue, or in the fibril form. And what you can see quite clearly is that the fibrils induce significant acceleration of the hydrolysis of a PNPA. We see much faster formation of the polynitrophenol product. So this experiment is actually quite important because if there is a catalytic reaction involving the fibrils only, we should have both the absorption of the substrate onto the fibril surface and the release of the product following the reaction or the interaction of the nucleophiles, in this case, lysine, serine, and tyrosine. Another experiment was designed to check whether indeed the interaction between beta amyloid and the PNPA substrate is responsible for the acceleration of the reaction. What you can see here is a FTIR spectra demonstrating that indeed we have fibrils in red, monomers in blue. Here you can see the sample of the fibrils that we obtained after 24 hours and which we did the experiment with. And the simple experiment here was actually keeping the same concentration of amyloid beta, and in these steps adding additional concentrations, additional samples of the substrate. And what you see is once we add substrate to the solution, after it reaches a plateau, we see an acceleration of the hydrolysis reaction. These steps are evidence that indeed the acceleration or the production of the polynitrophenol product is directly related to the interaction between the beta amyloid fibrils and the substrate. And so the conclusion for this part of experiment was indeed that the catalysts here were the fibrils, not the monomers. So we do have catalysis, and the catalysis is directly related to the presence of fibrils in the solution. Another very important question is, what is the contribution of the sequence or the different domains of beta amyloid in this catalysis? Okay, so we are asking ourselves, is it enough just to have fibrils forming from this particular peptide, or do we need also specific sequence, specific presence of amino acids that contribute to catalysis? So this question is actually quite important, because we don't want to have a situation where the mere presence of aggregates is inducing catalysis or is affecting catalysis. So in the experiment here, we took three fragments essentially of the beta amyloid. On the left, we took the complete sequence, the 142, that has the unordered component, the beta sheet, the loop, and the hydrophobic region. You can see it here. We also formed fibrils from the short sequence, short beta sheet forming sequence between 16 and 22. You see here also fibril formation due to the propensity of this region to form beta sheet. The third sequence included both the beta sheet region and the unordered end terminus of the beta amyloid. It also forms fibrils. Now, what about catalysis? The results were actually quite eye-opening. What you see here again is the formation of the product, the polynitrophenol product. What we see is that, well, I forgot to have the marks here, but the 142 beta amyloid, which is the red curve, is much more catalytic than both the A beta 1622 here in blue, or the alpha amyloid beta 122, which is in purple here. So this experiment tells us that it's not enough to just have peptide aggregates in that case. It's not enough to have the part of the beta amyloid peptide forming fibrils. We need to have the entire 1 to 42 sequence in order to affect catalysis. So it's not only the fibrillar formation, it's not only some formation of aggregates, but you need to have a certain region, amino acids, on the fibril surface that will affect catalysis. So we showed catalysis of beta amyloid with a model reaction, the PNPA hydrolysis. As I mentioned in the beginning, a more important question is whether catalysis by alpha amyloid beta fibrils are also responsible for the pathologies that we observe in Alzheimer's disease. And in this part of the research, we studied whether beta amyloid fibrils catalyze transformation or reactions of neurotransmitters. Degradation of neurotransmitters or destruction of neurotransmitters in the brain of Alzheimer's patients is actually a quite well-known phenomena. It's believed to be among the crucial side reactions of Alzheimer's disease. And it is believed that the imbalance of neurotransmitters is responsible for the cognitive damage of Alzheimer's disease patients. So, for example, there's been many studies reporting that changes in the concentrations of dopamine, adrenaline, acetylcholine, and serotonin, for example, these prominent neurotransmitters happen in the brains of Alzheimer's disease patients and might be associated or responsible for the severe cognitive damage of these patients. So the question we will ask ourselves, are amyloid beta fibrils responsible for degradation or catalyzed degradation of neurotransmitters? And in the following slides, I'm going to show you a few results, very interesting results, precisely demonstrating this point. So we start with acetylcholine. This is a well-known surrogate of acetylcholine, a very abundant neurotransmitter in the brain and also elsewhere. And what we see here, that again, the fibrils, beta amyloid fibrils, induce significant catalysis of oxidation of acetylcholine. The monomers have much smaller, quite experimentally insignificant effect. We see a similar phenomena in the case of adrenaline, okay? Adrenaline oxidation. Also here, you see that the fibrils induce a significant catalysis of oxidation of adrenaline, much more significant than either the monomer or the blank solution. Very interesting result we recorded in case of dopamine oxidation. Dopamine undergoes quite a complex oxidation process, and it ends up with a melanin or melanin derivatives. And the interesting thing with melanin is that in the brains of Alzheimer's disease, you find also melanin aggregates. That's a sort of a well-known pathological hallmark of Alzheimer's disease. So the results that we observed were, again, fibrils of beta amyloid induce oxidation of dopamine faster or more significant than the monomer or the control. And the interesting thing was that after addition of dopamine to amyloid beta fibrils, we could see these melanin aggregates that were highly similar to what people reported to observe actually in the brain. This table actually summarizes the enzymatic or catalytic activities of beta amyloid for three substrates, three reactions. Here, this is the oxidation of polynitrophenol. We see quite a fast uptake of the substrate onto the fibrils, release of the product after catalysis, and the enzymatic efficiencies. Now, the enzymatic efficiencies that you see here in these cases are orders of magnitude lower than enzymes. Okay, so it's not that the amyloid beta fibrils are fantastic enzymatic catalysts, but still, if you remember that the enzymatic activity of catalytic peptides reported in the literature is also quite small, between 0.3 and 0.7. These values are actually quite nice, and they indicate that indeed you have significant catalysis. These results were recently published in a paper in CAM Catalysis for those of you who want to look deeper into these results. So these are the results with beta amyloid. Now, obviously a bigger question would be if this phenomena, this observation of catalysis with physiological amyloids is something universal, or is maybe some kind of a coincidence or a quirk with beta amyloid. So here I'd like to show you some preliminary data that we observed with another peptide, physiological peptide called glucagon. Now, glucagon is a very important hormone. It has a role in sugar metabolism. It's primarily located in the liver and the pancreas. It's a relative of the well-known protein insulin that maintains a sugar balance in the body. The interesting thing with glucagon is that it also forms fibrils. It's something that's been known for several years now, that glucagon forms amyloid fibrils, quite ordered beta sheet fibrils. Nobody knows what these fibrils do. No, people found them primarily in a test tube, obviously, and they also identified in cells or in vivo, but the role or the function of these fibrils are not known. So the question we ask ourselves, do these fibrils have catalytic reactions? And what you see here are results in which we tested the paranitrophenol acetate oxidation and hydrolysis, the same model catalysis, catalytic reaction that we employed in the case of beta amyloid. And we tested a catalysis for the full-length glucagon, a glucagon sequence where histidine was removed. The reason is that histidine is a very well-known residue in enzymatic pockets in catalytic reactions of enzymes. We also use a peptide, a short peptide, which is only the hydrophobic motif of glucagon. We also tested a glucagon in which the hydrophobic motif was substituted by a sequence that prevents formation of fibrils. And we also tested a trout glucagon, which is the only glucagon isoform in nature that does not have the serine and histidine in the N-terminus. And these are the results. Here you can see again, quite clearly and beautifully, that the full-length glucagon is catalytic, highly catalytic. But when you start playing with the sequence, you can still see fibrils, both in the 229 and the 2027 peptides. They both form fibrils. Also, the trout glucagon form fibril. You can see it here. But these three sequences are not catalytic, or are much less catalytic. The glucagon that cannot form beta sheets, it cannot form fibrils. You can see it here. This sequence is not catalytic at all. So to summarize, this is basically the first report of natural amyloids exhibiting catalytic activities without cofactor addition, just the fibrils themselves, just the peptide. And what we observed for the first time that the beta amyloid can catalyze biological reactions that are or might be pertinent to pathologies of Alzheimer's disease, in this case, neurotransmitter degradation. Now, there's obviously lots of open questions, and most important of those is what is really responsible for the catalytic activity? Structure of the fibrils or the sequence of the amino acids? Most likely both. So what we're going to do next, we're going to do a further characterization of the catalysis products and the catalytic reactions. We need obviously to test other reactions and other substrate to see how general this phenomena is. And of course, it would be very interesting to expand into other amyloid systems. There's obviously lots of amyloids out there that are very interesting, and some of them we don't know much about their functions. And let me acknowledge the person that did all this work. This is my graduate student, Elad. And thank you.
Fantastic talk, Raz.
How am I in time?
Oh, perfect timing.
I'm pretty okay, yeah.
Yeah. So as we will have questions posted in the Q&A folder, I wanted to just start the question that maybe some of these are common in the Q&A as well. That can you give us a complete picture of which region of A beta is more important? Do you think the histidine residues are important in A beta as well or not? And did you try oligomers or protofibers or any other species and the mutants of A beta? Can you give us any details?
Yeah, so we, wait, I go back to the sequence. Yep. We believe that the, indeed, histidine is probably, likely actually, involving catalysis. Although in terms of organization, you have to have the hydrophobic and beta sheet regions to induce catalysis, to contribute to catalysis. But still, this is definitely an open question that we need to do more work. Regarding oligomers, you're welcome to go and read our paper, but oligomers are much less catalytic than the fibrils. So this catalysis phenomena is specifically related to fibrils, or primarily to fibrils.
What about any mutants of A beta? Did you try?
No, we did not try mutants of amyloid beta, except the several fragments, domains in the peptide. You can see 3 here. In the paper, we included a couple of more domains. They were all less catalytically active than the full-length fibril, even though most of them did form fibrils.
Okay, if we can look at the Q&A folder, there are many questions. Let's go from the top. We already answered the first question, are oligomers catalytic active? You already answered that question. Second question from Kuldeep is, what about oligomers in catalysis of acetylcholine neurotransmitter?
A very good question. I am pretty sure that the oligomers were also less catalytically active in case of the neurotransmitters. It's not 100% because I don't remember. You're welcome to check our paper, and if not, please write me an email and I will consult with my student, Elad, maybe he's in the audience, I don't know, and he could answer that.
Okay, next question from Chikun Liu. I think you answered the question. Do you have any thoughts on why the full length is more catalytic than fragments? You already answered them.
Definitely, because it's not enough to have fibrils. You need certain pockets on the fibril surface with distinct amino acids to have catalytic activity.
So can you say that in addition to the fibril structure, you need residues like histidine, right?
Absolutely. Yes,
okay.
It is likely a combination between the fibrillar structure or the fibrillar surface and the presence of certain specific amino acid that will form the catalytic pocket.
Our next question from Christian Grisinger regarding the catalysis of amyloids on neurotransmitters. What is known about the spatial distribution of the two? The substrate would have to meet its catalyst. Is this likely? That is, do released neurotransmitters diffuse sufficiently to meet amyloids? For intracellular amyloids, is it known that there is more metabolism of the neurotransmitters, which are mainly in synaptic vesicles and therefore not supposed to meet amyloids that are cytosolic?
Well, about the second part of the question, I really don't know. I don't know whether intracellular neurotransmitters would meet fibrils inside the cells, but this picture actually we took from a recent paper which specifically show interaction between neurotransmitters and amyloid beta in the synaptic distance, in the synaptic space. So in the extracellular milieu in the brain, it is certainly feasible that neurotransmitters will interact with beta amyloid fibrils.
Okay, next question from anonymous attendee. Thank you so much for the very interesting talk. So do you believe that fibrils could also be toxic indirectly by catalyzing the production of molecules that could be downstream toxic effects, example, invoking oxidative stress?
Well, this is absolutely a possibility. If we or other will prove that this is indeed the case, then you open here a new ways to understand Alzheimer pathologies. Absolutely.
So connected to that question, is it possible that during catalysis the fibers get disassembling scenario where you generate oligomers and more toxic species? That's also possible, right?
I don't know. I really don't know if that's the case. I think fibrils are pretty stable actually in the brain. I don't think that because of catalysis they will break down, although this is certainly a possibility.
But there are compounds known to destabilize the fiber structure.
Yeah, you're right. You're right. I don't know. Didn't think about this point.
Next question from Sheena Radford. Very interesting.
Hi, Sheena.
I noticed that the adrenaline oxidation data showed lag kinetics for oxidation. What causes the lag time? I also wondered if you had tried A-beta 40 fibrils for the DNP hydrolysis. Is it active and more or less than A-beta 42?
Actually, we really like this lag in the adrenaline oxidation because to me, I'll just show it here. To me, it says that the mechanism of the reaction, of the catalysis, is different between, for example, acetylcholine and adrenaline. And if indeed the mechanism is different, then it's an interesting observation because it can indicate that amyloid fibrils have different catalytic activities to different substrates in the brain, for example. Now, amyloid, we did not test amyloid beta 40, although obviously it would be very interesting to do. We directly jumped into amyloid beta 42 because of its perceived more significant physiological relevance.
Great. Our next question is from Carmelo La Rosa. Have you thought about doing these catalysis experiments with fibrils deposited on membrane? considering that biological reality membranes are present?
Great idea. We didn't do it yet. But yeah, certainly good idea.
Next question from Mark Font. Hello, congratulations for the excellent talk. What is the chemistry behind these catalytic activities? Do specific amino acids have specific catalytic activities?
Well, the answer is yes. It's not that amino acids by themselves have likely catalytic activity, but there are amino acids like histidine and serine that are well known to be located in catalytic pockets of enzymes. So it is likely that these amino acids are also involved in the catalytic sites on, if they exist, obviously, on the surface of the fibrils.
Okay, next question from Martin Michel. Do you see any effects of fibrils bundling on the catalytic activity?
No, we didn't test it. Again, it's a very good suggestion. Yeah, but we didn't test it.
So he's making a connection to Rolf Langen's seminar on individual fibrils being much more toxic than bundled fibers.
Beautiful, beautiful piece of information. Yeah.
The next question is from Jan Fischer. Nice work. Did you see specificity toward particular reactions that can be catalyzed? If yes, following which rules?
Well, we didn't screen so many reactions, actually. We did see that oxidation reaction, which I showed, is much more catalyzed by beta amyloid compared to other types of reaction. For example, dephosphorylation. That was another reaction that we tested. You know, the issue here is actually mostly technical, because there's only so many reactions that you can actually study, because you need markers. You need a certain fluorescent or visibly color-changing molecules in order to study catalysis. So what we are limited here in terms of screening large number of reactions is whether we have the assays. So in the sort of limited number of reactions that we tested, we did see propensity to oxidation or hydrolysis reactions.
Okay, let's go to the next question. Renato Dominguez, I think. When truncated peptides are prepared, it also changes the conformation of amyloid fibrils, as seen in the case of A-beta peptides that you showed. Did you find any correlation between catalysis and strain conformation?
We didn't go as deep, so deep. Again, this might be an issue. We really did not look into that. I have to admit that, you know, this work has been published only like 6 months ago, approximately. It's all obviously quite new. So there's many things to do. And these great questions will really make us do all these experiments.
There are a couple of more questions. Let's quickly go through them. Well, one is from Marina Lelli. It is possible that in vivo also metal ions play a role in the in vivo condition. That's what his question. So could they be coordinated at the surface enhancing the catalytic activity?
Very good question. Thank you. If you go to our paper, we actually found that when we added certain metal cations, well-known metal cations that act as cofactors in many catalytic reactions like copper and zinc, catalysis was actually inhibited somewhat, not completely, but partly, which by itself is very interesting observation. And it might actually indicate that in this phenomena is primarily related to the structure of the fibrils and the formation of catalytic sites on the fibrils, and the presence of cations might actually modulate the catalysis. So the contribution of metal cations definitely play a role. We're not sure yet how. And in the cells, whole new world. Yeah.
So there are three more questions. Maybe we can finish it. One from Beena Krishnan. Does the histidine side chain protonation status play a role in catalysis? Or have you tried pH dependence of catalysis?
Well, we did try pH dependence in the case of neurotransmitters. This is primarily actually in response to some reviewers' comment. There were changes actually. So the catalysis is actually quite sensitive to pH. This is something which is known in enzyme catalysis, obviously. You know that enzymes work usually in physiological pH the best. It certainly might be that the changes of the pH affect protonation of histidine, for example, which then affect catalysis. So that's certainly a possibility.
Next question from Danilo Milodi. Great talk, Raz. What about autocatalytic routes? May amyloid fibrils catalyze degradation of A beta monomers? Have you addressed this issue?
Great question. We haven't.
Okay, the last question, Penila Wittung. Would not the monomers start to aggregate to fibers within the experiment time of many hours? A beta aggregation is very fast.
Actually, the experiments that we did, first of all, in the fibrils, we waited 24 hours until we formed the fibrils and we observed catalysis. Now, in the case of monomers, we made sure that after 6 or 9 hours in the experimental conditions that we used, the concentration, temperature, there was not a significant formation of amyloids. However, it is certainly possible that, for example, in the slide here, that you do see monomers affecting some catalysis, and also we saw it in the case of the other catalytic reactions. When you wait 9 or 12 hours, you do have fibril formations which are responsible for catalysis. So the contribution of aggregation throughout the reaction might be a factor.
Okay, thank you very much, Ross, for the great talk and great.
Rams, can I ask a quick question?
Yeah, please go ahead. Yeah. Go
Okay, great talk. I really enjoyed it. I was curious to see, have you tried to look at the surface of the A beta 42 fibrils? I mean, the structure has been reported by several groups to see if you could actually identify the pocket or the residues responsible for catalysis.
In what, using what technique?
I mean, the structure was determined by solid-state NMR by 3 different groups, I believe, and it's remarkably similar. So it looks like A beta 42 has that structure. So you might try to look at the structure which is deposited and see whether you can identify something interesting on the surface, just by in silico, I mean.
Great question. Yeah. No, we did not do that. Yeah.
All right.
We took for granted that there are fibrils organized and have a certain structure on the surface.
Yeah, it's worth looking into the polymorphic fiber structures. Okay, so I would like to welcome Dr. Enrico Rivera. Please take over. Thank you very much, Raz.
Thank you, everybody, for the great questions. I need to do stop share.
The end, yeah.
Yeah. How do I do stop? Yeah.
So I thank Rams for this, for giving me the opportunity to introduce Claudio, which has been my supervisor basically for all my career. So I'm very happy to do so. Claudio got a degree in chemistry from the University of Florence in the group that later became the bio-inorganic chemistry group led by Professor Bertini. He was a postdoc in the University of Florence. Then I became a CNR researcher, and then a researcher at the University of Florence until I took quite early for the Italian standards a post of a full professor at the University of Bologna. And then he came back to Florence in 1996. He received several honors for his research, and I just list a few, starting from the Raffaello Nasini Gold Medal, which is awarded to under the say best under 40 researcher in inorganic chemistry, and then Federchimica Award for an Intelligent Future, the European Medal for of the Biological Inorganic Chemistry Society, the Gold Medal of the Italian NMR Discussion Group, and he was selected as ISMAR Fellow in 2016, and quite recently he was awarded the Richard Ernst Prize in Magnetic Resonance that happened in 2018. And now I think it's time to hand the floor to him. And please.
Okay, thank you, Enrico. And thanks, Rams, for inviting me. So the subject of today's, well, the title I selected was Molecular Aspects of Protein Aggregation. Of course, just a few aspects, but I thought if you put them together, you may find an interesting story. This is where I come from, which is the Magnetic Resonance Center of the University of Florence. And for those who do not know us, this is the center. And as you can see, there is a lot of NMR machines. So you can imagine that most of my talk will be based on NMR experiments. We also do, of course, other experiments with other techniques, but the main leitmotif of my talk will be magnetic resonance. I should say that this center is a node of the structural biology in strategic infrastructure. As you may know, in Europe, we are organized in research infrastructures, which work pretty well. So we are a node of structural biology, integrated structural biology. We do the NMR part, other people do X-ray, mass spec, and more recently, of course, cryo-EM. So the lab is surrounded by several other facilities, which we exploit to carry out our research. So the pathway towards fibrils. You heard a lot already from Raz about fibrils and what they can do. I will try to stress a few points along the formation of fibrils, along the pathway of formation of fibrils, starting from monomers to oligomers, which have been already touched by Raz, and the endpoint is, of course, mature fibrils. I stress this, and I actually would like to start from the endpoint because, because what we learn from mature fibrils is that, and this was mentioned by Rams, I think, in one of the questions that were asked to us, that they are often polymorphic. And polymorphism is something that seems to be really in the nature of fibrils, which means that the formation of mature fibrils depends a lot on what happens along the pathway, which it means in turn that formation of fibrils is a very complicated issue. And of course, it also means that along the pathway, there may be species that are more toxic than fibrils, and depending on how they form and how they then evolve into fibrils. These are just a few pictures of A-beta 40 because we started working in this field by looking at the structure of A-beta 40. And also there are many other structures. This is just to remind you that up today, probably the best way to look at the structure of fibrils is solid-state NMR, because X-ray has been successful in very few cases with very small fibrils. there is no way, or at least it seems to be very difficult to find structure to detect and solve structure of mature fibrils by X-ray, because they don't crystallize. It's very simple. Cryo-EM is making a lot of progress, and it's getting close to atomic resolution, but yet it's not there for complicated things like fibrils. So solid-state NMR is actually still the technique of choice. This is what we did back in 2011 about the structure of A beta 40. There are other structures of A beta 40 that are different from this one, and they are equally good or equally bad, because we actually don't know what is the dominant structure of A beta 40 fibrils in vivo. And I think the most successful attempts have been those of seeding fibrils by using fibrils from the brain of dead people. So it's a complicated issue. This is just one of the structures. It's a good one. It's very well resolved, but it's by no means the only one. So this is a view from top. Now, why I'm showing this? Because, well, you heard from Raz about A beta 42. And A beta 42 is, of course, very similar in sequence. It only has these 2 extra amino acids at the end. So you would guess that A beta 42 would form very similar, if not identical, fibrils to A beta 40. But this is not the case. In fact, A beta 42 escaped all attempts of ordered fibril formation until a few years ago. And then it was finally solved. And as you can see here, and this is the structure that I was suggesting to Raz to look closely, because Raz might be able to identify significant spots on the surface of this fibril that could be possible catalytic sites. But anyway, as you see, this is an S-shaped structure, whereas A beta 40 has a structure of C type. So it's completely different, even if the 2 sequences are identical, except for 2 extra residues here. And this has been solved in 3 different labs. So there is little doubt that this is the structure, the predominant structure. But it is at the same time a structure that it is very difficult to obtain. In most cases, A beta 42 aggregates very rapidly, and it forms amorphous aggregates. So having this structure is great, but we really don't know whether the toxicity of A beta 42 is linked to this mature fibril structure or to some of the oligomers. And then the most interesting issue is that in vivo, both A beta 40 and A beta 42 exist in different ratios. And it is known since many years now that if we increase the relative concentration of A beta 42, then formation of toxic species, I am not saying fibrils, of toxic species is actually favored. So several people thought that actually A beta 40 and A beta 42 could co-fibrillate, but this was never demonstrated. And in fact, there were evidence that they segregate rather than forming fibrils together. Several papers pointed to the fact that they tend to segregate, which is a little strange, because the fact that they coexist and that their presence is linked to toxicity would suggest that they could actually interact with one another. So what we did, we prepared mixtures of A beta 40 and A beta 42 in different ratios, and I spare all the details, and which were actually suggested. The experimental scheme was suggested by this paper, and some of the authors of this paper are actually co-authors of the paper that we have published a couple of years ago. Then we found a way to obtain fibrils from mixtures of A beta 42 and A beta 40 in a 1-to-1 ratio. And these fibrils, and what I was saying before about NMR, it's clearly evident here that just by fingerprinting the NMR spectra, you can see that the structure of A beta 40 and A beta 42 in these mixed fibrils are actually identical. And you can do that by labeling one of the two, for instance, A beta 42, and looking at the blue spectrum, or labeling A beta 40 in the mixture, so two different experiments, and seeing the red spectrum. And you see that there are very, very tiny differences, not significant differences, also in the NCA spectrum. So the conclusion is that they form fibrils together that are intertwined, and they mix in a 1-to-1 ratio, and they actually alternate 140, 142, 140, 142. So the difference being those two residues that are hanging out from A beta 42, the last two residues, are probably preventing the formation of the same type of fibrils when A beta 42 is left alone. But if it is, let's say, diluted by A beta 40, then this is a way for them to form mixed fibrils together. We think this is relevant to the understanding of the pathway of toxicity when, as it is happening in reality in sick people, when these two fragments are in a similar amount. So that's all I wanted to say about the endpoints. The message is that the endpoints are very variable. There is polymorphism all the time, and therefore we need to understand why this is happening. So we can start from monomers and before the onset of aggregation to see whether monomers bear with them some kind of okay, this should be an animation, but it's really not relevant, not so much relevant. This is an animation of alpha-synuclein, and you can tell that the protein is very disordered, and in fact, there is no sign of preformed structures in monomers. So you should not expect ever to see single beta strands forming randomly. This is not the case. And we have been able to show that by doing relaxometry, which is a specific technique that allows you to measure relaxation times as a function of field. And the shape of these dispersions, for those who are into NMR, those are spectral densities, in fact. And you can tell that the folded protein has a very high relaxation rate, above 1,000, whereas an unfolded protein like lysozyme at pH 3.5 has a factor of 10 smaller relaxation. And this is also happening for monomers of A53, 118, 108. This is all alpha-synuclein. Other proteins behave in the same way. And from the shape of these curves, it is very clear that there is nothing which is really preformed, except that this slight order that is residual there, it's less than 10%, is actually a phenomenon which is linked to the nature itself of the monomer, which of course reorients locally very fast, but also has some kind of global reorientation, which doesn't mean that there is a global structure at all, but the global reorientation accounts for this slight dispersion. You see, we published several papers, one of which was published in 2014 with Christian, which I think is in the audience today. And that was, we think, a very interesting paper in that it shows that there is no order except what is expected from any reptation movement of monomers. Other examples here, tau protein, lysozyme, and so on and so forth. So, endpoint is very variable, but it's not the fault of monomers. Monomers are monomers, and for truly disordered proteins, there is no sign of transient order. Nothing that we can detect, at least. So we are left with what happens in between, which is, of course, the main question for all the people who study amyloid aggregation, in fact. Aggregation kinetics followed by NMR. So our ambition would be to go from monomers to oligomers to fibrils and try to monitor this by NMR. As you know, there are several ways of monitoring the formation of fibrils, including fluorescence, TiO2, and so on and so forth. But this is molecular, and the information that you get from NMR is very, very deep, and NMR is really very rich of information. So how do we follow this phenomenon, even remembering that oligomers are probably the most interesting and most toxic species? So what is the problem here? The problem is that once monomers start to aggregate in oligomers, they all end up in fibrils. And monomers and oligomers are transient species, so they are actually difficult to follow. And even from the kinetic point of view, to detect the formation of oligomers on the way from monomers to fibrils, it's a challenge. So the trick that we played already several years ago was based on what we call sedimentation in the rotor. Now, for NMR experts, if you do solid state, you need the rotor that spins at the magic angle spinning and has to spin very fast. Now, a side effect of spinning a solution instead of spinning a solid in the rotor is that the solutes may actually sediment. The rotor acts as an ultracentrifuge. And so depending on the diameter of the rotor and the speed, the spinning speed, you may sediment solutes of different size. So we selected the rotor and the speed of the rotor in such a way as to sediment oligomers, but not monomers. So we start with a solution, clean solution of monomeric species. They could be A beta 40, they could be synuclein, and perhaps other species as well. We start spinning the rotor, and as soon as oligomers of a certain size are formed, they sediment on the wall of the rotor. And all of a sudden, they generate solid state signal, whereas the signal from the solution goes down. So this is a very good way to stop the oligomers before they can actually form fibrils, because you sediment them before they can actually evolve to fibrils. So by ultracentrifugation, you have to imagine that this is the wall of the rotor, and so the monomers will go through more and more concentrated solution until they are trapped in the sediment. Mind you, a sediment is not a precipitate. The sediment is something that is there, at least in our conditions, because of gravity. And if we stop spinning, the sediment comes back to solution. So a sediment is something that is reversible, and we have done it with proteins. We can sediment proteins and bring them back in solution by just changing the rotation speed. So we can study the oligomers, and we can study the kinetical formation of the oligomers. And you see, I think this is a beautiful picture because it's taken in the NMR rotor by alternating solid state experiments and solution experiments. Solution experiment, an inept, for instance, detects signals that come from the monomers only, because oligomers do not provide much signal at all. But once oligomers are formed, they are sedimented, so they behave like a solid, and they give rise to CP cross-polarization. And you see that those two curves are actually very symmetrical. So you see that by the time you decrease the amount of monomers, you increase the amount of oligomers. And you can also assess the kinetic, and we did that. And we had the model of formation and dissociation of oligomers. And the model is relatively simple because we don't have to worry about the successive formation of fibrils. So this was a very clean experiment, which allowed us to, for the first time, I think, to monitor the formation of oligomers in a situation that blocks the formation of fibrils. And we can also follow the aggregation kinetics by looking at the NMR signals of the monomers. And the NMR signals of the monomers are actually very easy to detect because monomeric A beta 40 or monomeric synuclein give rise to very sharp signals. And if you look carefully here, you see that there is a small decrease of intensity at the beginning, and then a sudden drop at this point. This is time. And all the signals more or less behave in the same way. So what's happening here? You can tell from here, you see that the cyan curve is the decrease in intensity of the signals of the monomer. You see that there is a slight decrease in intensity and then a sudden drop. And if you look at IoT or fluorescence or whatever, you see that this curve is also mirrored by this other curve. That is, you have formation of some protofibrils or oligomers that are still already sensitive to THT, and then formation of mature fibrils. So we can tell, and for the first time, we can actually address specifically this part of the time evolution, the formation of oligomers. We know that they are forming because we have the previous experiment from solid state, and now we see that they are actually present, and then they evolve into mature fibrils. So at this point, we have to insert into the kinetic equations all the terms that we need. And you can actually tell that if you look at the evolution of those signals and you try to fit the evolution of those signals so that the magenta and brown and green refer to different concentrations of monomers. So green is 30 micromolar, brown is 50 micromolar, and magenta is 100 micromolar. So of course, the magenta drops earlier because it's more concentrated, then comes the brown and then the green when the concentration is low. But you see that in all cases, if you fit it to a simple two-state model, two-species model, the fit is not very good at all. And this holds for both A beta 142 and A beta 40. But then if you actually include the formation of oligomers, then you get an almost perfect fit. Well, it's not really perfect here, but you can tell that there is a big difference with these other cases. So in our three-species model, you can actually account for the formation of oligomers and then for the formation of fibrils. And so you can define a full kinetic equation starting from monomers to oligomers, and you include an association, dissociation of monomers to the oligomers, and then oligomers that get together and convert into fibrils. Fibrils can still grow with the addition of monomers, or can even occasionally break into subfibrils, let's say. So all of these components are here. And by using these full equations, we have actually been able to account for the details of the time evolution of this system, which is A beta 40. But then how general is this? And we were, to our surprise, we were really happy to see that by looking at alpha-synuclein, we got a very, very similar behavior. So formation of oligomers and then evolution towards mature fibrils. And here we have actually taken advantage of an assay that's been developed not long ago, that is becoming actually a very good assay for clinics, because it's based on protein misfolded cyclic amplification, which means that you actually seed the formation of fibrils from biological fluids by adding small amounts of seeds. And this is very powerful because it amplifies the formation of fibrils, and so it's able to detect situations where fibrils are about to form. And particularly in the cerebrospinal fluid, this is actually the most reliable nowadays, the most reliable diagnostic result in detection of aggregates in CSF. Now, how can we exploit that? Well, we can actually do by fluorescence, by this new technique, what we have been doing by NMR. And guess what? At the beginning, we see an increase, which is increasing slowly, and then a sudden increase. So again, we know now that this is witnessing the formation of oligomers. So in a way, this is validating our equation because it shows to be actually very general. And the numbers are very reasonable. I mean, I don't have time to discuss all the parameters, but you can find them in our 2018 paper on A beta 40. And I hope you will find this for alpha-synuclein pretty soon. This is now submitted. But what was actually remarkable was that if we added some amounts, relatively small amounts of human CSF, then the curve is not the red curve, but it's the black curve. So apparently human CSF is inhibiting formation of fibrils. Why I'm saying fibrils? Because you see here that there is a growth. So apparently the formation of oligomers is not inhibited until a certain point. So this is now a guess, but the guess is that there is something in human CSF that is able to interact either with oligomers or with the oligomers that are actually turning into protofibrils. At that point, CSF is able to block everything. To get an idea of why this is happening, actually, this is happening, and it's even more interesting. This is happening also in the presence of preformed aggregates. So human CSF is actually able to revert the phenomenon and bring it back to monomers, actually. So what we did, we of course fitted this with an empirical equation, but which is related to our general kinetic equations that we have discussed a moment ago. So we have a first inflection here, formation of oligomers, and then a second one for the formation of fibrils. So you can actually have these A1 and A2 values as reporters of formation of oligomers and formation of fibrils, and the time it takes to get oligomers and then to get fibrils. And now what we did, of course, as chemists, we wanted to get closer to the molecular view of things. So we decided to actually fractionate CSF. And we made the pools starting from unfractionated CSF to a fraction which only keeps objects that are larger than 100 kilodalton. And then between 150, between 50 and 10, and so on and so forth. And what we found was very clear. Whole CSF was increasing T2 very much. This is actually out of scale here. And the same happens with the fraction with objects that are larger than 100 kilodaltons. All the other fractions, more or less within the error, behave like the pure buffer. So it's something here. And what do we have here? We have basically, well, there are many things, but the most abundant are HDL, LDL, and transthyretin. Now we can exclude that. So we took the three separately. Transthyretin produced a very weak effect, whereas both HDL and LDL perform more or less the same effect as the entire CSF. So it's something related to HDL and LDL. And this may not be surprising for those of you who remember that actually fibrils, and maybe even before fibrils, oligomers on the way to fibrils can actually be absorbed on membranes. And if you think of HDL and LDL, those are, you know, fat balls, more or less, with some proteins embedded. So this, we think, it's a very interesting result. To conclude, sedimentation allows us to focus on the monomer oligomer evolution only. And this was the experiment performed by sedimenting the protein into the NMR rotor itself. Our kinetic equations fully describe the monomer oligomer fibril evolution, and we have this as a starting point, which is very important. So we had only to extend that to include the formation of fibrils. And this holds both for A beta and for alpha-synuclein. The variety of types of fibrils must depend on subtle factors influencing the evolution of oligomers. It's not the monomers, and it's not the fibrils themselves. So it's on the oligomers. And what we found along this research was that CFS component, mostly HDL and LDL, inhibit fibril formation by sequestering oligomers. oligomers or protofibrils. It's sure that oligomers still form, but they don't progress to fibrils. So this is all I wanted to say. And okay, let's see. And I thank you for your attention.
So, thanks, Claudio, for your presentation. There are already some questions. Actually, one is anonymous attendee who is seemingly complaining that you didn't mention the more recent cryo-EM structures of fibrils, and I think we can dismiss this.
No, I mean, I know that there are cryo-EM structures and they're getting better and better. Still, I think that the atomic insight that you get from NMR is still an added value. May not be so in a couple of years from now, but.
Then there is a very nice question from Rams. He asks, intertwined fibers are only observed. Does your observation rule out the presence of A beta 40 or A beta 42 alone fibers?
Yes. I mean, we have tons of NMR experiments to demonstrate that, and I didn't want to bother the audience to show them. But yes, we do see crosstalk between nuclei in A beta 40 and nuclei in A beta 42. So they must be really intertwined.
So does this mean that your sample preparation condition is such that the affinity for interpeptide is much stronger than homopeptide interaction?
Yes, and we have guesses of why this is happening. And well, the short answer is that we know that A beta 40 can form the same type of fibers, let's say C-shaped fibers. A beta 42 may not be able to form the same type of fibers because of the two residues at the end that are bulky, and they probably provide some kind of steric hindrance. In fact, A beta 42 makes these S-shaped fibers. Totally different. By co-fibrilizing A beta 40 and A beta 42, we kind of dilute these two residues at the end of A beta 42 so that they fit well within the structure of the mature fibers. That is our idea. But for sure, when we put them together, we don't tell them to make mixed fibers, and we always get mixed fibers. So we don't get A beta 40 fibers, for instance, under the same conditions under which when A beta 40 is alone, we do get fibers. So
does this depend on peptide-peptide ratio, the population ratio?
No. So actually, there was this paper published in 2010 where they said that a 7:3 ratio was the most toxic. But in our hands, if we put them in 7:3 ratio, we find the mixed fibers plus the excess of A beta 40. So they really don't like to be separated. They want to form mixed fibers. So if we put them in the same ratio, 1:1, then we get a clean species.
Thank you.
Okay, then there is a question from Yijin Tang, who was asking how much sample is needed for one NMR test, and what is the difference for using solution and solid state NMR to influence the result?
Okay, so the amount, well, depends on the rotor, depends on whether you want to do solid state. You may need a few milligrams if you work with a reasonable size rotor, to maybe 1 or 2 milligrams if you work with a very small rotor. And of course, a very small rotor is more difficult to handle, but you can do that. For solution, more or less the amount is the same. And in fact, the experiment that I was showing on the sedimentation in the rotor starts from solution, and you can see a clean inept spectrum. And then you see formation of oligomers, and you see a CP cross polarization solid state type spectrum from the same amount of protein. So I would say a few milligrams is good for both. Of course, the conditions are different.
Then there is a question by Feng Ding. Are the oligomers actually the high density phase or droplets of liquid-liquid phase separation? Can NMR tell the difference?
Excuse me, because I cannot see the question. Can you repeat that?
The chat
Yes,
box.
sure. Sure. Are the oligomers actually the high density phase or droplets of liquid-liquid phase separation? Can NMR tell the difference?
Huh. Okay, I guess you mean the oligomers that are sedimented. The sediment is a sediment, so it's something that is a kind of gummy stuff that is by no means a solid and is not a precipitate. So I don't know if you can call it liquid phase, but well, it's a very viscous liquid, if anything.
Enrico, I promoted Feng Ding and Christine Grisinger to the panel, so they can
Yeah,
ask the question directly.
Thank
Okay,
you, Rams. So it's no, I'm not meaning those are precipitated, but rather, so there are many reports showing that the amyloid protein first they form this called phase-phase separation. They form this high density, still liquid phase, like a droplet. People call it droplet. I'm just wondering, they are actually not precipitating. They're still like suspended. They grow as time grows, like this called Oswald ripening, like in the colloidal science field. They actually can grow this droplet. So it's still, they're not highly condensed. They are probably barely touch each other and still stay as a random coil-like structure, but has some contact between monomers.
Okay, I think I understand what you say. So we actually have not looked at how these oligomers are when they are in solution. What we know is that the NMR signals of the monomer disappear. So these oligomers are not very big, but they are probably big enough to broaden the signals beyond detection or almost beyond detection. So they could be the droplets that you are talking about. When I was alluding to the experiments in the rotor, of course, then you add gravity. And so gravity makes those particles, whatever they are, to sediment. And so the density there is higher. So there is relatively little water in between. Actually, we have estimated that the amount of water in the sediment for a protein, not necessarily for a fibril or for an oligomer, the amount of water for a protein is similar to what you get in crystals of proteins. So maybe less than half the mass is water. So they're rather packed. I think this is not in contradiction. I think that if oligomers are forming these droplets that you say, then they would sediment and form a sediment that we see.
Thank you.
So in addition to that, can we say that your oligomers are beta sheet structured because they seem to be binding to the THT dye?
They do, but the intensity is much smaller.
And
so
what do you see in the chemical shift values? Do you see beta sheet structure or random?
No, we don't see them. we don't see them in solution. We see something in the solid state. Yeah. The quality of the solid state spectra is not nearly as good as the quality of mature fibrils because there is much less order. But they do form beta sheets. But probably the amount of beta sheets is not as much as it is in the mature fibrils, and they are disordered. So it's likely that TIOT cannot interact so well as it does with mature fibrils. But it fits with the idea that there are, I mean, we have evidence and there are beta sheets, even if they're not as abundant as in mature fibrils, and TIOT detects them.
Do you have a number for the size of the oligomer distribution size?
Yes, we estimated, I think, 30 monomers. Enrico, do you remember what was our estimate?
One was with 16, and then they appear to be multiples of that.
So 16, 32, yeah.
That's how the fit was done in the slide you showed.
Because we selected the rotor speed in such a way as to trap oligomers above this kind of threshold. Smaller particles would not sediment under those conditions.
Thank you. Christian, you.
Hi, Claudio.
Hi.
Thanks for the nice talk. What was kind of fascinating me was this inhibition of the aggregation in the presence of LDL and HDL in the CSF. And so I was wondering, do you know how big that concentration is, and also at which concentration are you doing the PMCA with the I guess added alpha-synuclein? Because I mean, if it is kind of sequestration or so, then one would probably expect that if one increases the alpha-synuclein concentration at some point in time, then the seeding would still work. Or so, and the other thing is, I mean, enigma to me is that normally lipids are considered to actually enhance aggregation of alpha-synuclein if they have the right composition of charges and so on. But here it seems that they are really inhibiting, and I'm trying to somehow reconcile that.
Okay. So the concentration of LDL, HDL in CSF is not very high, and we didn't make an attempt to increase that concentration. Actually, we added a relatively small amount of CSF to the cuvette where we were doing the experiment. So it was further diluted, and it was enough to inhibit alpha-synuclein. Now, alpha-synuclein is of the order, but again, Enrico, correct me if I remember wrong, of the order of some tens of micromolar, and which is the normal amount that is used in this type of assays. So we didn't do any significant change to the normal conditions for the assay. Now you ask an interesting question, but I cannot answer. What happens if we put more synuclein? I don't know. But I guess at some point you would saturate the capability of CSF to prevent aggregation. And so you would see formation of fibrils, but we have not done that. The contradiction is probably not a contradiction if you think that it can actually be, as you say, that HDL and LDL may actually favor the formation of these aggregates, but they would bind the aggregates. So it's not that they dissolve the aggregates. That could probably just remove the aggregates from the solution, if you see what I mean. So you are left with a solution that doesn't contain aggregates anymore, but still contains monomers.
I mean, at least in our experience, I mean, once oligomers are formed in the contact with lipids, of course, the HDL and LDL is probably not vesicles or so. I mean, I have to look exactly what they are. But normally, once you have kind of a template or so, then it continues aggregating, and that's why it's kind of intriguing.
But that bring to fibrils?
Yeah.
Okay. So then it's different, because here they actually prevent the formation of fibrils. I'm not saying that they dissolve the oligomers.
Yeah.
They could actually even favor the oligomers, but they do not progress to fibrils, at least in the type of experiments that I've shown.
I can send you the finding that we published last year. I just sent it to you.
Thank you.
Isn't it your THD data show the oligomer intensity goes down to 0, right? With the
Yeah.
So that means oligomers are gone. Is that right?
Gone or sequestered.
But
what we can say is that they are not accessible to THT anymore.
Or is it maybe it's not accessible because of the molecular crowding?
That could be.
I see.
There is another question from an anonymous attendee that is, have you tested the cross-seeding between different amyloidogenic proteins?
No, not yet. Not yet, but that would be interesting. In this experiment, I guess you mean.
Okay, so now we can open up the discussion for both the speakers. Raz, you can come back as well. People can ask questions to both the speakers.
I have a question to Claudio while we're waiting for people to come. I might have missed it in your talk, and it might be, perhaps I can see how technically it can be difficult, but have you tried to do TM of the sedimented oligomers?
To do what? To sedimented oligomers?
To visualize them by electron microscopy. I see how that can be difficult, but have you tried
you
it?
Would say in German, you would say jein. Yeah, we did. The results were not so exciting, but we did look at them. And by and large, yes, there is something that looks like small fibrils, but they are not they're not mature, if I may say. So the sediment is actually a way to block the evolution to mature fibrils. You stop at oligomers, and well, we tried to do cryo-EM, but we didn't get much.
And do you have an explanation, because it's in the sediment? I'm kind of trying to figure it out. In the sediment, the concentration, the local protein concentrations will be really, really high. Can you explain why it's not fibrilizing? Because it's a concentration dependent.
That is a very interesting question.
I need to understand. You get my point.
Yeah, absolutely. And we ask that question ourselves a lot. But our answer is that in order to form fibrils, which are rather ordered species, even only monodimensional order, you need room. You need to allow for small particles, be them oligomers or even monomers, to aggregate and make the fiber grow, right? In this very crowded environment, as you say, and as it is, in fact, these oligomers are oriented randomly, and they do not have much space to reorient in a way to actually make something which is more ordered than they are. So if I get your question right, the question is very interesting. We think, and I can offer you this explanation, that being so crowded, there is no way for oligomers to reorient in a way that can allow the formation of fibrils. I mean, as you know, fibrils can actually form in a better way or in a bad way, even from solutions. I mean, there is usually an optimal range of concentration where you get good fibrils. If it is too diluted, you may not get them. If it is too concentrated, you may actually get amorphous aggregates rather than fibrils. So it's a little bit, don't take me wrong, but it's a little bit like crystallization, in the sense that you need time for a solution to make good crystals. If the solution is too saturated from the beginning, then you get powder. You don't get crystals. So it's a little bit of the same concept. I think you need time and space to allow a fibril to grow. I don't know if this is
yeah.
convincing, but
No.
this is
I
what
was
we see.
actually keep on thinking in down that lines. I just don't want to take over. But if you, I'm kind of trying to translate it to the crowdedness of the environment, because what you're saying that in the sedimenting one is quite crowded. Have you compared it? How compares this to the actual cell environment? I know it's quite different because it's made of different things, but still, I mean, is it similar?
Yes, it is. It is. I mean, we have many examples of proteins that sediment in this way, either by ultracentrifugation simply, or even by doing them in the rotor. So we know what is the crowdedness of proteins, folded proteins, and they are more or less in the same ballpark as the crystals of the same proteins when you allow them to crystallize. So they are crowded, and the amount of water is of the order of maybe 50% or even less. So the same happens for fibrils. So you can, not for fibrils, sorry, for the oligomers, disordered oligomers. So they are as crowded as proteins in a crystal or proteins in a sediment. This we know.
Thank you so much. I just I can give my.
So Claudia, I have a question, technical question. In your inept versus CP experiment, both are done under Major-Congo sample spinning.
Yes.
So if you adjust the delays in inept, you should be able to see the signal where the transfer via dipolar coupling would occur between proton and X nuclei. Did you try that?
No, I think that was a simple inept experiment to detect the stuff which is in solution. So basically monomers. And we didn't attempt to detect by solution techniques the oligomers, because they sediment, and so, and it's a very clear sign of the formation of this sediment, the fact that you start seeing the CP. If you start with monomers, you don't see anything.
But you said signal to noise ratio is poor in CP. Well, so why do you think it's poor?
No, did I say that? No. No.
I asked a question, is the chemical sheet beta sheet like? You said it was not clear, something like that.
No, I mean, the spectra are very bad quality. They are visible, but they are not as clean and as resolved as are the spectra of fibrils in the solid state. No, there is a lot of signal. So
they're not clean means they're broadened or crowded?
Yes.
That you're more than one conformation. Is that possible?
Absolutely. You have a whole range of conformations.
So that's an
but you can tell that there are beta structures from the chemical shift, but the resolution is
well, you cannot quantify it because of resolution.
No.
Yeah. Great. So it looks like there are no other questions. Great talks. I really enjoyed it very much. Thank you both very much for coming and giving excellent talks and staying back for Q&A. Thanks, Claudio. Thanks, Enrico. Thanks, Raz. Thanks, Christian.
Thank you, Rams. Thanks a lot.
Thank you.
Bye-bye.
Thank you.